All right, everyone. Thank you so much for joining us today for our Build to Sell webinar. We are excited to listen to today's talk. And with that, I will turn it over to Aleksei.
All right. Yeah, thanks so much for the invitation. I must say I was a bit surprised because I don't really work on synthetic cells yet, but that is an inspiration for me. So we'll do more of those. So as I was thinking about what to show you today, I realized that over the course of years, my lab has been working in the general field of uh, building machines using synthetic components. And one specific area is building machines that are membrane bound. So in biology, you have a cell has a membrane and lots of membrane proteins. They do all kind of things. And I just realized that we actually have worked quite a bit on that. So that's what I will talk about today. Just very briefly, other things that we do in our lab, we are interested, I would say we specialize in high-end simulations of biomolecular systems. We are interested in building complete biophysical models of biological cells, small organelles, reconstructing the three-dimensional structures of packaged viruses, biological condensates, artificial water and ion channels, nanopore sensing and sequencing, and DNA origami. So DNA origami is actually something that I'll talk a lot today. But before I do that, let me introduce the method that we use. It's all atom and multi-resolution microdynamics. We call it the computational microscope. So the idea is that we build atom by atom microscopic models of various systems and then animate them in time by solving Newton's equations of motion. So at the end of the day, we get something that looks like that, which is um, uh, a movie of something going through a nanopore. So DNA origami is the method invented by Paul Rosamond in 2006. The idea is that we can build anything out of DNA by folding a long strand of DNA into a predefined shape using an ensemble of staple strands. So here you're looking at two pieces at viral genome that will be brought together by a staple strand that will hybridize. And there's a short movie that illustrates how you would potentially build a logo of the University of Illinois by self-assembly. So the method works really nicely. Here's some examples of designs that were put forward and also how they turn out in experiment. Yeah, so my journey into DNA origami field started with a quest to build membrane pores out of DNA. And our original motivation was to use it for all kind of sensing and sequencing applications. But it turned out that they are actually of interest of themselves. So a cell membrane is full of all kind of membrane-bound entities. So you have channels, transporters, antiporters, and so have you. Now, DNA generally is thought to be a molecule that avoids being inserted into the lipid membranes because it is hydrophilic and the interior of the lipid membrane is hydrophobic. But it turns out that if you add enough hydrophobic parts to a DNA construct, you can facilitate its insertion into lipid membrane. So, for example, in this pioneering work by Fritz Zimmel and Henrik Dietz, they added a bunch of cholesterol anchors here to a DNA origami construct, and that just inserted into the lipid membrane. In parallel, Stefan Haworka's lab realized DNA nanopore insertion by changing the backbone of the DNA to make it to basically to kill its negative charge, and that worked. Other methods have been proposed, differing by what kind of molecules are used as hydrophobic anchors for the DNA. So let me use this system to illustrate how our computational methods work. In experiment, you characterize DNA nanopores by first embedding them into vesicles and then placing them on a nanocapillary. So once they're placed in a nanocapillary, you apply electric field using electrodes and you measure currents that flow through it, so you get a current versus voltage curve. Now we can do exactly that in our computational microscope. We start with the design. We relax the design, add cholesterol anchors, place the whole thing in a lipid membrane, add typically magnesium and some other salt, water, and boom. So we have an all-atom representation of experimental system. Then we can apply electric field and see how ions move through it. So we can measure directly the current from the displacement of ions, and we can do it at multiple applied voltages. So at the end of the day, we kind of have a complete account of what's going on in this microscopic system. So we can measure how much charge is transported versus time. So the derivative of that or slope of each curve would be the average current. We also know where exactly the ions go because we can make streamline plots and so forth. So at the end of the day, we get current versus voltage curve, which can be directly compared to current versus voltage curve that is experimentally obtained. And that's kind of validates the method. Uh, all right, so what did we learn? In one of our earlier simulations, we were looking at a DNA pore made of four DNA helices with cholesterol anchors. And we were specifically wondering what happens to the lipid bilayer membrane. So when we set up the simulation, the lipid bilayer was flat, right? So those are lipid head groups, so they're all kind of positioned parallel to one another. But as the simulation progresses, the lipid membrane changed its shape, so it kind of curved, and which is schematically shown here. So really, the lipid head groups, because they're hydrophobic, they kind of uh, form a toroidal pore that surrounds the DNA nanostructure. And that actually was not a great news for the usage of DNA pores for sensing because, for example, for this pore, most of the current flows outside of the DNA, not through the central channel. But that kind of gave us an idea that maybe we can build a membrane channel that doesn't have a channel, right? So if we just take a molecule of DNA and add enough hydrophobic anchors, then it will form this toroidal pore and the ions would just go around it. So that's something that was experimentally realized by Kirsten Goffrich when she was still back at Ehrlich Kaiser Lab at Cambridge. So here's a simulation that shows how the design works. We start with a double-stranded parts, also porphyrin anchors, and right away you see that there's a water-filled pore surrounding the DNA, and if you apply electric field, ions would move through it. Now, we can measure directly the current as I described and compare it to experiment. It's basically right on for this kind of a construct. So we have realized a pore, a DNA, a membrane channel that doesn't really have a channel in the biomolecule. Now, our second example from that era has to do with transport into nucleus. So we were interested in designing larger pores for delivery into cells. So something like that, where you just bring a syringe and inject things into it. Our early work in this area was targeting the establishing the DNA construct. So here's the syringe that we've built, and again, this was experiment done by Ulrich Kaiser lab. It's a big system, 112 helices, but it still inserts into the lipid membrane. And if we look at the conductance, we're getting around 13 nanosiemens, which is similar to the conductance of a nuclear pore complex. Coincidentally, I guess. Yeah. So we can build small things, we can build large things. One thing that was kind of came about from the simulation is the following, and that's actually one of the examples which I often give when people ask me, can we predict anything? So here's what we predicted. So you're looking at an image of a DNA nanostructure embedded in the lipid bilayer. So as previously described, you'll have this nice curved toroidal pore. So the consequence of having that toroidal pore is that now lipids can spontaneously transport from one leaflet of the membrane to the other, because now there is this passage, right? So without the DNA, it's not there, but with the DNA, you have this continuous passage, meaning that the lipids can diffuse freely from one leaflet to the other. That is actually what specialized group of membrane proteins do. They are called scramblases. And all biologically relevant membranes, they have asymmetric composition because typically what's inside a cell or an organelle is different from what's outside, and you need specialized lipids to interact with the cell machinery. So the scramblases, they are there to rapidly equalize the composition of the lipid bilayer membrane at some certain conditions, just by that. And there are all kinds of medical factors that are associated with misfunction of the scramblases. So in our simulations, we've shown that a DNA nanopore actually is a very, very efficient scramblase. It, in fact, is maybe thousands, tens of thousands times more efficient than any known biological enzyme. And it took about 2 years for a very talented graduate student to actually demonstrate that that is the case in experiment. And the way it's done is using the assay that biologists use to probe for scramblase activity. So very briefly, you have a phospholipid that is labeled, and then you add a quenching agent called dithionite, and that will kill the fluorescence. So now if you have a DNA scramblase construct inserted, then the fluorescence should go away to 0 because both leaflets uh, of the membrane will eventually become exposed to dithionite, and if you have a non-scrambling construct, it just goes to half, and that's exactly what they saw in experiment. But more excitedly, a consequence of that is that it marks the cell for programmed death, so-called apoptosis. And the reason is that typically on the inner leaflet of a cell membrane, you have so-called PS lipids. And if we insert the DNA scramblase into a cell membrane, those PS lipids will become exposed to the outside, which will be recognized by the cell machinery that something is wrong with the cell, and that cell will be marked for destruction. And that's exactly what experiments have shown. So among many membrane embedded things that are there, the earlier ones that we tried to mimic were so-called voltage sensing channels. So of course, they are essential for neuronal activities, and typically in biological system involve the motion of the charge. So the charge is a voltage sensor. So our early work in this area was together with Ulrich Kaiser and Philip Tunefel labs, where we used DNA origami construct to read out the voltage by measuring the distance between two fluorescence dyes. So here are the simulations that illustrates how it works. We have a nanopipette and a DNA origami plate with two fluorescent dyes. But the spacing between the dyes will depend on the voltage that we apply, and that's kind of what was demonstrated in this paper. Our later work in this area was to probe the membrane potential and actually in a cell membrane. And there, the idea was that we take a DNA origami plate and we add again two fluorophores. And because one of them contains kind of a charge linker, if you apply electric field, then the distance between the two will change. So this actually took a while to figure out why it works, because experimentally it was shown to work. But the mechanism that came out, it is actually not what we originally planned, because inside of a lipid membrane, you typically have a electrostatic potential that is much higher than the outside. So really what this voltage sensor senses is the effect of the membrane potential on the local concentration of ions. Again, the mechanism is kind of complicated, but it's described in this paper that you can have a look if you're interested in the subject. Of course, one of perhaps the most spectacular membrane-embedded machine that is known to man is FOF1 ATP synthase, right? So that's the protein complex that synthesizes the majority of ATP that is used to fuel all kind of biological processes. So the way it works is you have a membrane part that converts a gradient of uh, electrical gradient and chemical gradient of uh, proton species into torque. And then the torque is propagated to the water-soluble part that catalyzes ATP synthesis by producing the cyclic rotation in the soluble part. So we've been trying to construct synthetic systems that have some kind of a functionality of a FOF1 ATP synthase. So this one was a collaboration with Henrik Dietz' lab. Actually, so his lab designed this construct. The key insight here was to make a DNA origami that is intrinsically frustrated. So there are like these pedals and a camshaft, and that camshaft is such that there's always, one of those pedals is unhappy because there's something that sticks out there. And the idea was that if we kind of rotate the thing, then we will cyclically change the conformational changes in the structure of this origami. Yeah, so actually making those systems was complicated, and Henrik is one of the, if not the expert in this, so it's really amazing what they can build with DNA origami technology. So our role here was to probe what happened if we actually apply torque to this construct, whether or not we'll indeed see the cyclic changes in the conformations, and that's exactly what we saw. You can kind of see, so as we rotate the central part, that this kind of camshaft would induce cyclic conformational changes in the rest of the origami. So that's kind of where we were with this experimentally and simulation wise. We haven't been able to actually show the synthesis of ATP, which is of course very difficult, but it's something that someone will eventually do, I hope. But meanwhile, we were also interested in this membrane-bound part, right? Because it is, in a way, an electromotor. Um, we are all familiar with electromotors. Some of you may be driving them. But the way they work is that you have a coil of wires and magnets, and then by loss of induction, you generate torque, right? So this kind of a mechanism is very difficult to scale down because of the physical constraint. The way this motor operates is it relies on volume forces, but as you shrink things down, then surface forces dominates. Even at the scale of sub-millimeters, you already need to resolve to different physics, like this electrostatic ratchet. Um, and were some efforts in the field to engineer rotary apparatus. So one was again from Henry Dietz, which showed that you can assemble something that will undergo rotational diffusion. There was a work from Fritz Zimmer lab where they kind of had a robotic arms that was like moving around in response to the changing direction of the flow, which was under the field. And this pioneering work from Carlo Montemagno lab shows that you can combine the soluble part of a 4F1 ATP synthase with a metallic post and to actually see how it spins. It's a really fascinating things. So we've been trying to build designs according to microscopic systems. So one of our earlier construct was this water mill, but it actually never worked because we didn't really understand at that time the physics of the DNA origami plates and how permeable they are. So basically, they are like very permeable. It's like they're not there because they're supercharged and there are ions around them. So when you apply electric fields, they are just super leaky. The other designs that we tried were these propellers, which were just too floppy. And this one actually worked, but no one could build it experimentally because it was just not DNA origami compatible. But then we thought, well, maybe, DNA is a chiral. Perhaps it can work as a electromotor. And that's what we set out to try here. So the way we do it is we took a fragment of DNA, we added water, ions, and then we did a little trick here. We so we restrain the DNA so it doesn't fly away in response to electric field because the DNA is negatively charged, but it could rotate left or right depending on what happens in the system freely. And then we apply electric field and watch what happens. And this is what happens. The DNA actually rotates. And the way it rotates is kind of what you would expect naively, if you would have, according to its chirality. Like if you take a normal screw and you would think, what would happen if you put it in a stream of water, it would rotate like that. So if we plot the angle of rotation versus time, it will just go to one. Um, Pretty steady state rotation. If we change the direction of the field, it rotates the other way. And most importantly, if you don't apply any field, it doesn't rotate deterministically, it just undergoes a stochastic rotary motion. Now, what else is chiral? So RNA molecules, they are also chiral, and double-stranded RNA also rotates in our hands, but maybe not so fast as double-stranded DNA, which we actually understand completely why, because it has a slightly different shape. It has a deeper grooves and a different charge density. So it all adds up that it rotates, but not so fast. The thing that really convinced me that it's a real fact was this one. So in a computer and also in a lab, you can build so-called LDNA. So it's like a chiral mirror of image of normal DNA. So the difference is only the chirality, and LDNA rotates just the same speed as normal DNA is just in opposite direction, right? So that was very nice. And the thing that of notice is the actual speed of rotation, which is billions revolutions per minute. So I would argue that you've made the fastest electromotor known. Now, why does it rotate? So the mechanism has to do with the fact that the DNA is a highly charged molecule. So it carries one electron charge at each phosphate group, so one electron per nucleotide. But the charge has to be compensated by ions that are dissolved in solution. So the ions tend to uh, form a cloud around the biomolecule, but they're not really stuck. They're still in the fluid, which means that the fluid is charged. So when we apply electric field, the charged fluid would flow, and this motion of the flow would impart impulse at the grooves of the DNA, and that will produce the rotation. So this is kind of messy plot that show that that mechanism actually exists, but here's a kind of a zoomed out view. You have a water flow, it imparts torque, so there's a reaction force, and that generates torque. Now, um, how would you actually realize something like that in the practice, right? Because previously I just showed you a DNA molecule that was in solution, right? So for all practical means, you would have to embed it into a membrane, and this is actually why it's part of the talk. So in our kind of first test simulations, we had a membrane that is made out of carbon. And again, we use this tricks that we restrain the DNA molecule to rotate to the surface of a cylinder. And if you apply electric field, it would spin, just as in bulk solution. The velocity will be slightly different because the velocity of water is different. But still it rotates almost a billion revolution per minute under these conditions. It's interesting to take a note of the energy efficiency, so we know how much power we input into our system because we apply electric field, and we know how much friction is there to rotate the DNA molecule, so we can divide one by another and get energy efficiency of about 0.1%, which is actually, you would think, pretty small, but it's not bad because it operates at the scale where fluctuations dominate any deterministic motion. So the fact that it still works is kind of amazing. And the final example in this area was this one. So we wanted to show that one can build a system, a realistic system that can be that can be realized in experiment and observe the rotation of the DNA. So in this particular set of computation experiments, we were not applying those restraining potentials or anything like that. Instead, we used a T-bar DNA. So this DNA is made of three strands. They form double helix here and double helix there. One essential component of the design are those unpaired bases, which stick to the surface of the membrane and prevent this whole construct from leaving the nanopore. So you'll see in this movie, when we apply electric field, you can kind of see how the DNA actually wants to leave, but it can't because it's tethered to the membrane by this hydrophobic attachment. And yeah, under 400 millivolts, we will get about 60 million RPM, still very, very fast. All right, so can someone realize this in experiment? So unfortunately, making those really, really tiny pores in where it's a fusion of carbon nanotube with graphene is difficult. One can make those, but at a slightly larger scale. So while we're waiting for improvement in experimental fabrication capabilities, we explored an alternative design, and that's was a wonderful collaboration with Henrik Dietz and Kees Dekker's lab. Um, it actually turned out that we worked on the same kind of design of biological, not DNA motors in parallel. And when we realized that, we saw that, well, let's collaborate. And so that's a design that was put forward by Henrik Dietz's lab, where it's not a single DNA molecule. It's an assembly of DNA helices that have the shape of a turbine. So Case Decker's lab has amazing expertise in solid-state nanopores. It's one of the person credited with their invention. And in this kind of setup, you have an array of pores. They're pretty big, 50 nanometers in diameters. And now to observe whether or not we generate the DNA turbine, we need to first arrest the transport through the pore. So we do it by using this very thick bar. And then somewhere here we place a fluorophore so we can actually see if it rotates uh, in a microscope. So in simulations, it rotates as you would expect for a turbine. And here's like a short movie that illustrates how it works in experiments. So you can see the constant kind of flies in and then it gets trapped, and by eye you can tell that it spins. There is actually a lot that went into this paper, and I don't have time to explain it, but surprisingly, the direction of rotation could be reversed by changing ion condition. That was totally unexpected. It took 2 years to figure out, but we figured it out. It's all in the paper. Now, the last thing that I want to tell you is that, well, with FOF1 ATP synthase, it is actually a reversible motor. So in a synthesis, under synthesis condition, it converts electrochemical gradient into synthesis of ATP, but in some conditions, you actually want to do it in reverse. It would hydrolyze ATP here and pump ions from against the concentration gradient. So we're wondering if if maybe our DNA is like that. In fact, could DNA be an Archimedean screw? So an Archimedean screw is this ancient device that people use to transport water against concentration gradients. You have some kind of a spiral system. You crank it, and then you get water flux. So here's kind of an impression of how it would look like. We have a DNA molecule again in a membrane, and then we apply torque, and we see if water and ions flow. So let's see. So here's our setup. It's a similar setup to what I showed you for demonstrating DNA electromotor. The difference is that now we are actually mechanically rotating DNA, and we're seeing what happens to water and ions. So it looks like that. We apply torque, so it spins. The velocity with which it spins is proportional to the torque that we apply. But more importantly, we can measure how much charge is transported through it, and we clearly see deterministic transport of charge, meaning that if we change the direction of rotation, the charge would go the other way, and so on. So here's a movie that actually illustrates the process. So you're looking at three ions, and they are being transported down here. So I mean, there's a lot of stochasticity in this process, but eventually we'll see how, like for example, the blue guy actually escapes. Yeah. Oh no, it goes up. Yeah. So for under these conditions, we are getting approximately half an ion transported on average per one turn of a helix. Um, and chlorides are excluded from the nanopore environment, so that's why transport of chlorine is almost negligible. We can convert this into current, and it will give you about like 30 picoamps, something that you can actually measure in experiment if you could build something like that. So similar things happen with water. If you rotate the helix, water is also being transported. And to give you an idea how much water is transported, it's about 10 water molecules per revolution of a helix. So if you want to transport it, the entire volume of this nanopore, it would take about 100 revolutions. Now, but can it actually act as a pump, right? So that was really the question. To demonstrate that, we set up a system where we had a different concentration of ions above and below nanopore. So that took a while to figure out how to do, but now we know. And the idea is now that we have a DNA molecule and we will apply torque to it. And we hope that we can pump ions from this low concentration compartment to a high concentration compartment because of the action of a helix. So in the absence of torque, right, we have a concentration gradient, meaning that we would have uh, potassium ions diffusing through down this concentration gradient. Why potassium? Because the DNA is negatively charged, so that makes the transport selective. And of course, we would have also a water flux, right? So you'll have water flowing along with the potassium. Now, as we start rotating it, we're getting less and less transport, and at some point, at some kind of a speed, we get pumping action, which is illustrated by the movie here on the left. Now it's interesting to compare this to FOF1 ATPase again. So to generate a rotation in this range, we have to apply a torque of about 50 piconewton nanometer. That is exactly the ballpark of torque that the soluble part of ATP synthase applies under synthesis conditions. So this is actually very reasonable torques. Now, the energy efficiency again is pretty poor. So here again, we know how much mechanical energy we put in, and we know how much electrical or hydrostatic work we produce. So if we combine the two, it's getting about fraction of a percent, which is a lot less than the numbers that are quoted for biological motors. In fact, it wasn't all that straightforward to get numbers for FOF1 ATP synthase working in the pumping regime. There are a lot of work claiming 100% efficiency, but that's usually for how well the hydrolysis is transformed into rotary motion, but into pumping it's a little bit more complicated. But numbers, you can find numbers around 60%, and this is a lot less, right? So you could say, oh, this is bad. Actually, it's actually not bad because we are transporting 10 to the 8 ions per second, right? And that's about, well, 5 to 6 orders magnitude more than ATP synthase transports under pumping conditions. So really, I would say that our design trades efficiency for throughput, and and that's how we kind of sell it at this point. All right, this is pretty much all I wanted to tell you. Thanks so much for your attention. This is my lab at some point. So the key people to highlight: Jijung, he was a postdoc who started DNA origami work in my lab, and Chris developed all kind of models for DNA origami, and he was the person who realized this DNA electromotor. And Xavier did the last part of the talk where he showed the DNA can act as an Archimedes screw. Thanks so much.
Great. Thank you for that wonderful talk. There are questions in the chat. If you could read them back so that people who watch the recording can then.
Yeah, let me see if I can do it. Okay, so there's some sound issues.
After that.
So when you generate channels using DNA hydrophobic compounds in lipid membrane, does the type of molecule passing through this channel ions affect the interactions between DNA and lipid membrane? Does it affect efficient? I mean, it's a great question. I assume that we could have some multivalent ions that would close the gap, between the DNA and the lipid membrane, especially if the lipid has a certain type of head groups there. We have not systematically investigated, but I would assume that is definitely something that can happen. Yeah. Okay. That was the question from, okay, now the question is from Henry Taylor. You demonstrate that changing your mechanical stiffness of the stator can modulate radial rotation of the camshaft Do you think this represents genuine mechanical energy transduction, or is it primarily a modulation of the rotor's diffusion landscape? Yeah, that's a great question. So again, we in that work we have not demonstrated synthesis. It's very difficult to do with the current origami technology because it just doesn't have precision to place molecules in the proximity that is required to catalyze synthesis. So that's kind of really a fundamental challenge, and but labs are working to overcome it by making origami stiffer. No, but really the purpose of that work was to show that one can build a system with this intrinsic asymmetry and produce cyclic changes in the conformation through rotation of the rotor. I hope I answered the question. All right. If you wanted to deploy an electrical motor machine like this inside a synthetic cell, what would you consider the main limitation? Yeah. Which do you think would be biggest bottleneck? Fueling, controlling mechanical state, or preventing interactions with messy intracellular environment? This is a great question. Of course, what we want to do is we want to build a system just like what I've showed you, but the one that embeds into lipid membrane, so you can actually place it in a synthetic cell, maybe use it as a kind of a flagellum rotor, synthetic, and things like that. Yeah, that's a really exciting direction for us to work, and there are several experimental labs for pursuing this. What are the main limitations? Um, I think just assembling the thing is pretty difficult, and then for the thing to work, you need to have additional means to generate the electric potential, right? So we can convert electric potential into torque, but that electric potential has to come from somewhere. So you also need to have a way to sustain the gradient of either electrical gradient or the chemical gradient, and that has to be done in parallel with embedding the rotor. So that's, I think, is kind of difficult to do at the same time. Messy cellular environment. I don't know. I don't think that would be the main showstopper at this time. Okay, Trisha. The rotation is attributed to electrosmotic flow around the chiral DNA. How do you distinguish the torque generated by electrosmotic flow from a torque arising directly from the interaction of the charged DNA with electric field? Okay. I mean, what is the minimal physics ingredient that makes the system a turbine rather than simply an electrical driven charged molecule? Oh yeah, that's a really, really good question. So one thing, like first to note is that if we just have the DNA and no ions, no water, it doesn't rotate because there's nothing for it to produce torque. It's negatively charged. Okay, fine. So it will just, accelerate an electric field. You need to have a fluid that would interact with this with the chiral aspect of the molecule, right? So if you don't have a fluid, then the DNA itself doesn't know that it's chiral in the electric field. Now, we did a study where we looked what actually contributes most. Is it the ions that are bouncing off the backbone of the DNA, or is it the water molecule that is drugged by ions? And yeah, it is the water molecules, because we could realize a system where we had kind of similar physics, but only ions, right? So they would have the proper friction coefficients and everything, and then we would see rotations 5 times faster because the transport of momentum from ions to DNA is more efficient than through water. But in reality, you always have to deal with hydrated water, hydrated ions, and that's why it is essentially just like a turbine where you have a water flux that generates torque. Although the water flux is generated by electrosmotic effect. Yeah, that's a great question. Since the system is driven continuously out of equilibrium by electric field, do you consider the efficiency on converting electrical energy into mechanical work? How is the torque rotation relationship changes as you increase the load, and yeah. No, yeah, that's a great question. I kind of partially answered it in my talk, but right. So just more. So when we apply electric field, we also know how much current we produce, and that's how we estimate the input power. And for the mechanical work, it's just we knew how much work is dissipated when we rotate the helix because we computed separately the rotational friction coefficient, and from those two you can get the numbers. But everything is linear, so you have to realize that everything is linear in the system in a way. So if we increase the electric field, then it rotates proportionally faster, so it doesn't really change energy efficiency. Uh, for the DNA pump work, we also looked at how the diameter of the pore affects energy efficiency. And there, interestingly, we found a maximum. So it's when the pore is like a nanometer larger than the DNA, then the energy efficiency has its maximum. And then it kind of drops in either direction, actually. It was pretty interesting that if you make the pore like really small, so it's barely like the DNA can barely fit. It's actually also not great because you don't get any water flux in that case. So, yeah, I think I answered all questions.
Yes. Well, if there are no more questions, thanks again for a great talk and have a great day, everyone else.
Yeah, thanks so much. Bye
Thank
bye.
you.